Professor Terrence Deacon, Professor Michael Levin, this is a long time coming. I think many of the listeners and viewers of the show have been waiting to see this conversation. Both of you have such distinguished careers and have massively shaped and influenced developmental evolutionary biology and cognitive science as a whole. And you have so many overlapping areas of expertise and common interests. With that being said, Mike, perhaps I could start with you. What do you think of Terry's work over the years, and which aspects of his work do you find most fascinating? And then Terry, I'll let you answer the same question when Mike is done.
Yeah, look, I have read your stuff since the 90s, the developmental neuroscience papers, especially the cross-species neural transplants and things like that. I thought it was incredibly interesting. Since then, I mean, there's been some amazing developments that you've led. The incomplete nature and the focus on the origin of and the importance of semiosis, I think, is absolutely critical. And all your ideas about the tearing down the binary kind of Cartesian dichotomies. I mean, I'm all on board with that for sure. The flows between form and function. I mean, there's I could go on and on, but all of this merger of these deep ideas between philosophy, evolutionary biology, developmental biology, I find absolutely critical for progress. So thank you for doing all that.
In many respects, those are exactly the things that are exciting about your work, and in particular, where we overlap very strongly is this recognition of, in a sense, the deep commonalities that are underlying everything that's biological, including the informational part, but also the deeper than evo-devo, in the sense that it actually utilizes sort of the self-organizing logic that we find in all kinds of systems that are really critical pieces of development, of evolution, of cognition. Having spent a good part of my career looking at how brains develop, for example, seeing that a lot of similarities are involved in the development of, for example, planaria, a lot of your work on planaria that shows a lot of that similar sort of pattern generation stuff that goes on that is not strictly biological. It's certainly not designed in any obvious sense. It's a very different kind of logic, and that search for that underlying logic, which is, I think, where we overlap so strongly.
Yeah,
and of course, the very fact that you had specific time to work with Dan Dennett, who had a significant influence on me while I was in Boston. He and I have had these conversations back and forth over the years on different sides of the same issue, oftentimes sort of not agreeing, but recognizing, exactly how these different perspectives inform each other. And I think that's where you and I will probably fit, because we have a common interest, but very different backgrounds in terms of the particular topics we've worked on specifically.
Yeah. And Dan talked about your work a lot,
I mean, with that being said, what are you guys' thoughts on Dan's work and his legacy in philosophy, and how's it impacted both of you?
Why don't you go first, Mike, since you've been working with him directly, or you worked with him directly?
For me, there's a few things. He was always to me, he was an incredible example of somebody who wanted to understand things in a fundamental way that was, the things he said about that there is no philosophy-free science. There's only, what do you say? There's only science where you smuggle in, philosophical assumptions without examination, right? And so that, I think, was very critical, and his commitment both to really clear, concise, rigorous philosophy and the data, the experimental work that is necessary to revise your philosophical outlook is very important, because I find, in our field, I find a lot of the times when I give these talks that are partially this kind of philosophical stuff and partially data, and some people say, okay, the data are great, the experiment's very good, don't talk, like, all this philosophy stuff, you don't need it, don't talk about it, and I said, well, why do you think we did those experiments? We wouldn't have done them otherwise. That's the whole point, right? And I think, especially in some of the molecular biology approaches and things like that, people tend to feel, or at least they act as if they're really touching reality directly. There is no philosophy underlying their perspective. They think that the things I say are metaphorical, but pathways and things like that are real. We got real things. We got these pathways, and just I always liked Dan's kind of unflinching fusion of the philosophy and the data.
I would say that's exactly the same for me. That sense that he's a philosopher that's totally focused on the science, the technology, that it always influenced his thinking. It always is very important. And as a result, he was coming largely in particularly the 80s and 90s, when we spent a lot of time interacting. His inputs were from computer science and cognitive science, as done particularly from the MIT groups, and mine, of course, was from the biology side. And we'd go back and forth on this. I remember he used to have regular meetings at his home, and we'd sit around and argue over these things. And he was always in the middle. He was always in a sense recognizing, well, listen to each other. That kind of thing. Just really, really helpful. And yet was after some of these really basic philosophical questions. The free will question was a big part of his thinking. The idea of experience, of consciousness, that everybody sort of gave him a hard time for coming up with a title, Consciousness Explained. That wasn't what he was about. He was actually about interrogating the problem. Why do we think the way we do? And as a result, even though he and I came to very different conclusions about that, at least during those years, it was just a really constructive discussion. And I find, like with Michael, so I began, I should say, just to be clear, my career was driven initially by the discovery of a philosopher. In the late 1970s, I read the work of Charles Sanders Peirce, mostly by accident. I was thinking I was going to go on and do, just sort of standard bench science. And the philosophy just sort of kicked me hard, really on, that he was asking questions at the end of the 19th century, the beginning of the 20th century, that nobody was even asking now, 75, 80 years later. And so when I began to do my neuroscience and evolutionary work, it was always driven by this sort of deep philosophical question that sort of drove it about, the nature of representation of information and the processes that generated it. So in that respect, I always had, like Michael, in many respects, this sort of a philosophical grounding that kept forcing me to ask these questions. Now, wait a minute, it's not that simple. It's just something more.
Mm. Well, beautifully said, guys. I mean, rest in peace to Dan. This conversation has so many overlapping themes, well, the two of you and your work, that it was very difficult to actually decide on what to ask the both of you. But I managed to set a few cards together. I think let's start off with a deep one, origin of purpose. At what point, Terry, perhaps you could start. At what point do physical systems, without brains, let's say, begin to exhibit purpose-like behavior?
Well, I actually approach this in a very different sense. That as I realized that even simple organisms like bacteria were way too complex to actually sort that out. And so I've actually tried to simplify it as much as I could. And I've approached it mostly from a thermodynamics point of view. One of the things that I've never felt comfortable with, and yet it's sort of the standard philosophical approach, is a list of properties. What's the list of properties that's going to define life? And as we've seen over the course of decades and decades and decades, that list of properties, it always leads you to not have a definitive answer. And so what I wanted to ask was, what is it about the thermodynamics of life that's very unusual? And because when we look at, and I think what happened is that over the course, I think it begins partially with this book, What is Life? A series of lectures that the physicist Erwin Schrödinger produces in the 1940s. He kicks us off into asking the question, why does life acts so different than other systems. He recognizes that there's an informational issue, but in particularly recognizes there's a thermodynamic issue. And I came to believe, partly because of Peirce's work, but also because of later work, that in effect, the thermodynamic question and the information question were the same question. The problem is that I think really beginning in the early 1980s and on until the present, many people, particularly approaching this physically, have been satisfied with the far from equilibrium. I describe it as morphodynamic, but a lot of people call it self-organizational processes. That's all you have to really cover, because according to the Schrödinger perspective, the really interesting question about life was that it's far from equilibrium and it keeps itself there, and it produces form rather than destroying form. It produces regularities rather than having regularities spontaneously break down, like the second law would have suggested. What I came to realize, and really, as I was struggling with how brains develop and why language is so unusual for communication amongst other species, that in effect, there's a third transition, that we can't just stop and looking at how form gets generated. We have to think about processes in which form gets remembered. It gets generated and remembered and repaired, so that in effect, if it gets modified back again to where it was. It's sort of like a memory that gets passed on. And that wasn't answered here. And so I wrote this book in the mid-90s, began this book, and then it doesn't get done until a decade later, that came out to be Incomplete Nature, which was basically to say, no, the problem of emergence, first of all, we got to get rid of the magic. It's not magic. It's about novelty of some kind, and typically it's combinatorial novelty in an interesting way. And what I realized is that what's happened is that we have oversimplified the thermodynamic story, and that we needed to have a thermodynamics in which self-organizing processes themselves organized each other in such a way that they rebalanced and then had something like memory. And so that I really saw the origins of life question and the origins of interpretation, not the origins of information, because I think we have oversimplified the concept of information. We flattened it so that we don't really deal with the question of evaluation. We don't really deal with the question of reference, how something present can be about something that's not there. But that there's an organization, a thermodynamic organization, that makes that possible. And therefore, it's not a surprise that some of the mathematics of thermodynamics and the mathematics of information have a lot in common. Even though they're talking about very different things, they have a lot in common because there's some similar processes. So to just cut to the end of that story, I realized that the problem that everybody has in biology is with viruses. Are viruses alive or are they dead? And I realized that the question about being alive was not the right question. It's the question about this contortion of thermodynamics that allows something to be about something, allows something to make predictions about the future and to then maintain those predictions. How can a chemical system do that? So the question always dogged me. I actually wrote a paper with this sort of title, is that how does a molecule become about something that it's not? That it just doesn't have that. We just can't assume that DNA and RNA are just information. They're about something. They're not just copying themselves. They're about something, and they're about themselves in an interesting sort of twisted way. So I realized that the question of viruses was really the crucial question. Viruses are at that boundary where we're not sure we can say it's alive or not, and yet it does all the things that living things do. That is, it evolves, it protects itself. We can talk about, killing viruses. All those things we can do, even though we know that it's not quite the same language. Mm-hmm. So I've sort of, in the last decade of my career, I've been focusing on that question, which I think is a way to ask the question about what is information in all of its various forms, what I would call the semiotic concept of information, and then how it comes about and how it evolves.
Mike,
so that's a long answer to your question.
That's fine, Terry. Mike, same question. At what point do physical systems exhibit purpose-like behavior? But also, at this point, you could also address whatever you think that Terry said is important, valuable, and where you might disagree or agree.
Right.
Yeah, there's not much to argue with there. I agree with almost all of it. So maybe I'll just talk about a slightly different perspective on some of these things that I've been developing. So what I'm interested in, I think a lot of times dichotomies lead us astray. So trying to, is it alive? Isn't it alive? Is it cognitive? Isn't it? Like, I think these things land us in these pseudo problems that are probably unresolvable. And so what I'm really interested in are models of scaling and transformation. So as Terry said, was saying, I want to know, how do the things that we're interested in, so the ability to generate form, to maintain it, to have this memory, to all of these things, how do they scale up from the tiniest beginnings? And so that's one thing that I'm really interested in, is the scaling. And the second thing is this idea, when I think biology does this really well, is polycomputing. So this is something that Josh Bongard and I have been developing, this idea that what you really have, instead of one objective, correct, this set of physical events is computing this function, or this has information about this thing, is to consider all the different observers. And so within a living structure, you have many nested observers, right? You have both laterally and vertically. You have the system's parts. You have the other systems around it that are all continuously interpreting each other and what each other are doing. And because they're all hacking each other, right? They're all trying to figure out, where do I end? Where does the outside world begin? What are the things I have control over? What can I affect? What are the most efficient signals I can get it to do other things, and so on. And so this idea of, and of course, systems also, as you said, systems also have to interpret themselves. That's the most interesting thing about significant agents, is that it's not just that others apply a sort of intentional stance to them and see goal-directed behavior, but they themselves are able to close that strange loop and do it for themselves. So this is the sort of thing that we've been working on, is these stories of scaling, which has many implications for cancer and for other things. But one thing in particular that I'm really interested in is the concept of mistakes. So as you observe chemistry doesn't make mistakes. Chemistry just sort of does what chemistry does. But developmental biology can certainly make mistakes, and behavior can make mistakes. And so the extent to which you have a system that, and this is how I see all of these things, as navigating different spaces. So of course you have metabolic spaces and physiological spaces and transcriptional spaces, and then anatomical morphous space, which is sort of what I study mostly. And then you have 3D motion, space of motion, and so on. As systems navigate this space, it is very natural to look at that scale, that the Wiener-Rosenbluth-Bigelow scale from passive matter to sort of active matter, and then how much what are the tools that you can bring to understand the navigation of the system in that space? Is it a random walk? Does it do delayed gratification? Does it have memory and forward planning? Does it what are all the things that it does? And this and so what we've been doing is taking tools that are usually deployed all across, maybe let's say, the right side of that spectrum. So tools from computational neuroscience and behavior science and so on, and asking whether they apply to very simple things in other spaces that are really not easy for us to sort of visualize. And that's been incredibly enriching, because it turns out that if you're willing to do that, if you're willing to say that this is an empirical project, we can't just assume that this is how cognitive different things are. You have to actually do the experiments. When you do the experiments, you find out some amazing things, even at the very left end of that spectrum. So, for example, gene regulatory networks, not the cell, not all the stuff that goes in it, but just the mathematics of a few nodes linked to each other by the ordinary differential equations. Just that alone can do 6 different kinds of learning: dynamical system learning, including Pavlovian conditioning, including habituation, sensitization. They can count to small numbers, things like that. So you get that sort of for free, so to speak, from the math right at the very beginning. And when we talk about purpose, you can sort of define, purpose versus goals, right? Do you have a purpose? I mean, that's kind of a metacognitive, sort of thing. But the ability to navigate that space using the tools that we associate with some level of intelligence, I think, goes all the way down. We see it in extremely minimal systems. We've even done. And my background is originally computer science, and so I like very simple kind of algorithmic things. And it turns out that even in very simple deterministic things like sorting algorithms and so on, you can find surprises that look like different kinds of problem solving that you have no business to expect from the algorithm that you thought you wrote. So yeah, to me, these things start very low down, and we just have to do experiments to see if we can find them.
Yeah, the way I like to think about that transition is the transition from chemistry to normative chemistry. Chemistry in which there is good chemistry or bad chemistry, or chemistry that works and doesn't work. The question is, for something to be normative, there has to be a beneficiary. Something has to benefit or be harmed in the process, to do better or do worse. So that it's the critical transition. And for me, in some respects, I'm at the very bottom and the top, because I was very interested in brains, and particularly uniqueness of human brains and their anatomy and function. And then also it forced me to the very bottom, ask the question, well, when can you say it starts? When is there finally a beneficiary? When can I now say something actually benefits, as opposed to just being preserved like a rock is preserved, or generated like a chemical reaction generates more of something? None of those have beneficiaries, in the sense that a beneficiary is something that, in a sense, does work to maintain itself. And that's an interesting question. So work comes part of it. This is why it's necessarily thermodynamics, but why I also force myself to think about the simplest possible systems, simpler than a virus, because viruses are parasitic and they're taking advantage of lots of stuff. But basically, virus-like structure has a lot to it that has this kind of, you can think about it as just I don't think about it algorithmically, but certainly in terms of it has something like self. Whenever we talk about living systems, we talk about self-regeneration, self-repair, self-reproduction, self-correction, all of those things. We've got this notion of self. I've often thought that this is one of the critical problems in philosophy of mind, is that we just sort of assume self. Self is actually a really complicated issue, even though it might be very simple. It's actually something that if we just leave alone, we just assume a beneficiary, we just assume goals, and don't talk about how they're generated, I think we get into circles.
Yeah, I mean, something very interesting about this concept of beneficiary is also related to valence and reward and punishment. So we can all see that you can reward and punish a paramecium, simple enough. But then you can ask yourself, what does it actually, if I started backwards and I say to people, okay, can I punish a chemical network? And they say, well, absolutely not. And then you show them paramecium. Well, what's this? Because you can do that here, right? And what we're finding that's really interesting, and this paper will be out in, I don't know, hopefully soon. If you have a model of a pathway or a gene regulatory network, so not the rest of the cell, no evolution, nothing, just a small set of nodes that are connected to turn each other on and off. In addition to the learning that we find there, you can also find something very interesting, which is that if you take metrics from causal information theory that people are using to apply to brains. So people like Giulio Tononi and Eric Hole and others are applying these things to try to understand, am I looking at a pile of neurons, or is there somebody home in there? Is there a person, is there a human patient that's in there, right? So it turns out that not only do some of these networks have significant causal emergence, but that causal emergence goes up after you train them. So you can calculate it. And so what happens is, for some of them, not for all of them, there are classes. In fact, there are, I think, 5 distinct personalities that these networks fall into. But for some of them, as you start to train them, and when I say train, I mean you stimulate some of the nodes. So let's say in a Pavlovian paradigm, you've got your unconditioned stimulus, your conditioned stimulus, and then there's some other node that's your response node. And so you start to pair the stimulations, and then you see what happens to the response. So as you do these things, for some of them, the causal emergence goes up. And so you've got this really interesting loop where by virtue of learning about their environment, in this case being trained, not so much learning, more being trained, they become more integrated as a tiny little self. They become, right, they acquire this kind of integration. And for some of these things, what we looked at biological networks versus random networks, and so it's very interesting. The biological networks are better at this, significantly better at this. They do more memories, they do more causal emergence. So clearly, evolution, I think, liked this feature, but even the random ones do it a little bit, and so that suggests to me that even before you get into any kind of selection or any kind of evolutionary cycles, just from the math alone, you get this little free gift that kickstarts some of this stuff, and then of course you can optimize the heck out of it, as you do these selection cycles. But right at the beginning, just the math that governs these kind of networks. And of course, some of it, Stu Kaufman talked about some of this stuff a long time ago. But it's not just the dynamics. It's not just the dynamical system. It actually has both learning ability and this kind of cohesive integration that grows as a result of experience. So I think we can trace, and as you said, both very sort of very high and very low. There is this kind of weird cycle where you can take metrics from the kinds of things that neuroscientists do with human patients and look at how this works with very simple systems.
So what I was going to add to this is something that we've actually done at the other end, that is the neuroscience end. I have a couple of colleagues at the University of San Francisco in which we're looking at simultaneous EEG and fMRI and asking the question, because my assumption in all of this is that the dynamics, or what you might want to call it computing, I'd rather call it semiosis, but basically the informational processes are generating the physical, material, energetic processes that make the information processes possible. That is, there's what Hofstadter would have called a strange loop in which the dynamics and the semiotic activity is generating and maintaining the physicality that makes it possible. So you get this loop between sort of you might say the ontology and the epistemology of a system in which each depend on each other. Well, we thought that must be possible also for how brains work. That we've been ignoring this in part because we assumed that fMRI was telling us that when these neurons get information and they're active, they change their metabolism. What we've begun to discover is that there's, depending on the task, sometimes the metabolism anticipates and precedes the neural activity, and vice versa. And you can look at it in terms of different parts of the nervous system, in general, sort of these broad distinctions like a default network and things like that, but also between brainstem, midbrain, and forebrain, and so on and so forth. There's this interesting exchange that it looks as though a lot of what we would describe as self-generated activity, intrinsically generated activity, the metabolism precedes, and it drives up neural activity, which changes the metabolism, which drives up the neural activity, and then shifts metabolism to other parts of the brain. So that in effect there is both a leading and following feature in which you might say the computing or the semiotic activity, that is a signal generation activity, and its substrate are critically linked. And it turns out they're linked at a number of levels, not just neurotransmitters, but also things like nitric oxide and various ionic channels that sort of ionic potentials that sort of pass through large areas. All of these areas are linked so that there's no clear distinction between what you might call the information processing and its physicality. It's all part of the same thing, and that's sort of the other side, the other extreme, I think, of what you've been talking about, Michael.
Yeah.
You guys were talking about causal emergence. I'm going to do my best to just be a fly on the wall of this conversation, by the way. So, if you guys have any questions that you want to ask each other directly, by all means. But let's talk about constraint and causality. Michael, let's start with you. How do constraints shape outcomes more powerfully than initial conditions, let's say? For example, is absence more causal than presence?
Yeah. Well, I mean, the first thing is I'm completely on board with things like absences being causal, functional inputs into everything that happens. So I am on board with that. And I think I think Terry will say probably the most about it. What I would like to say is that in addition to constraints, I tend to track two other things that are kind of in that class. One is this interesting aspect that was really highlighted by morphological computing, where Pfeiffer and Bongard and folks like that, where when you have embodiment, and I study embodiment not just in the three-dimensional world, but embodiment in these other spaces, and I think it actually applies perfectly well to all these other problem spaces that life works in. There are many constraints that guide what's going to happen, but there are also another thing that's kind of amazing is that in addition to the constraints, there are also these ways that you can offload an awful lot of computation onto the environment. It's like, and this is what they show when they have these robots and other things, that basically there is no controller that has to handle all the things that has to happen. They're basically letting the environment do the compute for them. And this is something very new in our lab that we're just getting into, is asking, what does that look like in these other spaces? What does it look like to offload transcriptional problem solving, physiological problem solving, anatomical problem solving to the rules of these other spaces that absolutely provide constraints that will guide subsequent events? But in addition to the constraints, they're also helping out. I mean, that's the part that I think is really, really wild, is that there's actually all kinds of benefits that come from that where the environment is actually helping you do the thing that you want it to do. And the other thing that I track a lot are patterns that arise not from physical facts. In other words, there are mathematical structures, there are laws of computation, laws of geometry, of number theories, all these things that evolution, I think, uses a lot. It uses them as in effect free lunches, because you can save an awful lot of evolutionary compute time if you can just be handed certain truths from the laws of mathematics and computation. And I think those are especially interesting because they're not. Many of them are not determined by physical facts. They wouldn't change if you shuffle the constants at the Big Bang and things like that. And so those kinds of causes that are not the traditional, why did this happen? Oh, because the BMP, molecule is, well, that some of these causes are not like that at all. They're like a completely different class of things. And what's really critical for us now is because we try to drive a lot of these things into very practical programs in regenerative medicine and in bioengineering and so on, is to how to take advantage of these things. So we would like to know, as we look for causes that are beyond physics and history are the things that biologists love as causes, but then as we see, there are these other things, and being able to exploit those and exploit that space of these patterns that can be used for our construction and can help us communicate our goals to cells and tissues in regenerative contexts. So yeah, constraints and various other things that are like them.
There's so much in this question. And we'll go back and forth on it maybe in this process. But one of the things that sort of drove my thinking about this originally and pushed me back into the theory was an interest in the Fibonacci spirals of plants. And the precise mathematical relationships, these wonderful spirals that you see in, for example, the seeds of a sunflower. And knowing that the genetics was not doing the math, that the genetics was in effect only really controlling thresholds in which various regions of the plant was releasing auxins, these various growth hormones in the plant, and yet producing these remarkable spirals that we find everywhere. And recognizing that one of the advantages of these spirals is that if it goes up the length of the stem, for example, for producing leaves, then the leaves are maximally out of each other's way. If you want to capture sunlight really well, what you want to have is things that are just, they're as tight as they can be, but maximally also out of each other's way. And it turns out that it's wonderful that evolution would want this. But it didn't have to, in a sense, encode it because it was there in the geometry itself. And all you have to do is you want to, create different kinds of spirals, different kinds of Fibonacci's with tighter or looser spirals. You just change the thresholds in which things respond. And so at one point in time, somebody said that I should call this the lazy gene hypothesis, that genes will only control what they have to control. That, in a sense, if they are over controlling something, it's going to eventually degenerate if there's a redundant capacity out there, if that redundancy is produced by the constraints, in this case, of geometry of space. That caught my attention. And in fact, it's now been part of a recent work that I've been doing that I call inverse Darwinism, as we begin to look at evolution. I mean, this was recognized back as early as 1970. That a lot of genetic evolution is by virtue of duplication. But it's not just gene duplication, it's duplication at lots of levels. And one of the things that has to happen for evolution for life is that you have to always overproduce. You can't just keep up with the second law of thermodynamics. You've got to go a little bit faster. So you've got to generate too much. And that allows you two things. The redundancy provides, in information theory, redundancy is to correct errors. But redundancy in biology is to make you error tolerant. That is, if you have redundancy, then things can go wrong and you can still keep going. Well, if you've got a system that always overproduces stuff, that always makes redundant cells, redundant genes, redundant proteins, and whatever, you've always got a little error protection there. You've got a lot of little plasticity, which means you've got exploratory space available to you. Well, it turns out that in evolutionary theory, of course, we're thinking only of the eliminative side of things. But in fact, what we're talking about is that the generative side of things is not just generating random variation. It's generating redundancy. And redundancy has a very, very special role, and it plays a critical role in information theory. Now, this will bring me back to the question of constraint. Because when we look at the original mathematical theory of communication that Claude Shannon puts together, there are two measures of information. They both involve what he calls entropy, which is just basically a way to talk about the possible variety of something, either in time or in space or whatever. But there's the original entropy of what could have been produced. And then there's the entropy of, what results. So if you're sending a message, there might be a bunch of messages you could send, but the one that's picked up does not have all that variety in it. And so in effect, there's two entropies for Shannon, and mostly we focus on one of them, which is just what he calls the entropy of a channel. But the entropy of a message is the difference between what the channel carries and what was actually received. And that tells you that what's happened is there's been a constraint imposed upon that information. And that information was carried on something physical. But because it's something physical, it's subject to thermodynamic laws as well. And this is one of the reasons why the thermodynamic story and the information story are linked, and they're linked precisely by this concept of constraint. That is the received information that's a constraint on the possible information, in effect, is the result that there were something had to act on the signal. Something had to act on it to constrain its thermodynamics, which then would constrain its information entropy. And that tells you this is how it could be about something, because the constraint of the channel can now be, in a sense, about work that was done on it. Because to shift something to a less probable state takes work. And this then helps us understand the whole concept of work, which is, of course, necessary for everything we're talking about here. That it's not just the release of energy, it's the release of energy in a constrained context. You've got to constrain the energy release. So, even a piston in an automobile engine, where you constrain the explosion, allows you to push something far from equilibrium, to push a car uphill, for example. So that constraint is a necessary but oftentimes ignored or just sort of assumed part of work when we talk about biological systems. But if you recognize from information theory that it's the constraints that carry the information, but constraints are also what determines the structure of the work that was done. And the structure of the work can generate new constraints. So if you can generate new kinds of work with constraints, and that work can generate new kinds of constraints, you generate new kinds of work. The whole possibility of evolution is the result of this interesting relationship between constraint and the release and the control of energy. And that means that if you have a system that can both generate and then remember and transmit constraint, now you have a system that can evolve.
Sticking with that, let's talk about regeneration memory. You'll notice that the first question I asked, well, this previous one was more tailored towards your work, Terrence, but this next one's more towards yours, Mike. Perhaps, Terry, you could start with this answer regarding regeneration and memory. When a planaria regrows its head and retains memories, where do you think those memories are stored? And what does this tell us about the body as memory substrate?
It's interesting because memory assumes a lot of what we've talked about already. And it assumes one thing that we don't talk about a lot, but I think is very characteristic in planaria. It's characteristic in how brains develop in multicelled bodies and that sort of thing. And that is that you can initiate the system, start it going with a highly compressed source of information. DNA is compression. And so when we talk about complexity, one of the problems we want to talk about is, you got a list of numbers. Is that list compressible in the sense that there's some redundancy in it? If it is, you can take advantage of that redundancy in reconstructing from a compressed version. And this is where algorithms oftentimes play a critical role. And in fact, we've analyzed complexity these days since Chaitin and Kolmogorov and others, in terms of what I would call a decompression rule. A decompression rule is you've compressed something, and now you got a decompression rule. The key is what is the decompression process? It's not that all the information is stored in the DNA. What you've got is a highly compressed system that if you pair it with a decompression system, whether it's algorithmic or it's a set of constraints that are built into the geometry of the world or built into the recursive activity of some process, then you can take advantage of. This is that lazy gene idea that I was talking about. That in effect, what we want to talk about is that whatever you can rely on in your decompression process, you can now leave out of the compressed source. And so, this is even true for something as trivial as, architectural design. For the architect knows that there's going to be wood and metal and tools and people putting it together. Knows that somebody will know how to decompress this model and do something with it. The decompression is ignored because he can just assume it's there. That's the problem, is that we tend to ignore the decompression process. And so what I like to say in terms of my favorite example of this is a frog built out more complex than if you've taken the body of the frog and thrown it into a blender and blended up into a mix. In one sense, the mix is much more complex. You would have a hard time coming up with a systematic set of algorithms to figure out where different kinds of molecules were located. But in the adult frog, it's much more simple in one sense. So it confuses our concept. It's simple in the fact that if I know that there's a bone cell here, I know there's likely to be a bone cell next to it. If there's a skin cell here, you don't have that in the frog smoothie. You basically don't have any predictability, and yet at that level, it's more complex. But it's a misunderstanding of complexity, because what's actually happened in the case of the actual frog is that by taking a highly compressed representation that is its genome, in a decompression system, its cell that now depends upon other cells, their position, what they release, what their electrical ionic activity is, how diffusion processes when they release molecules will affect cells close by versus those different from them, and so on. All of that's part of the decompression process. But the decompression process is not to be confused with the compressed representation. And so I've been very much interested in two ways. The decompression process is the functional part, but in fact, evolution has to work backwards. It has to figure out compressions. And I think one of the real problems we have is that we tend to think of it only in one way. We look at the function of something and say, no, let me just now sort of break it down into its parts and its function. No, it evolved backwards in some sense. The compression comes second. So I've always told people that, this view that DNA or RNA came first is a mistake. That was a compression of some other dynamical process. And once we understand the decompression process and how the two have to, in a sense, evolve together, we realize that things get better and better at compressing and decompressing. And that's the complexity issue. But it's a pairing of those two processes.
Yeah. So super important. This compression decompression thing is critical, because one of the kind of meta constraints that biology has is that the substrate is unreliable. In other words, on a large scale, you're guaranteed you're going to be mutated. On a small scale, you don't know how many copies of your proteins you have, that things are being degraded. And that's quite different than all of the computational architectures we make now, where you work very hard to have abstraction layers, where if you work at a high level language, you don't worry about your copper of your registers floating off and so on. And so I think one of the implications of that, if and we've done a lot of simulations on this now, that evolving over an unreliable medium. So there's two components that I'm really interested in. One is the unreliable medium, and the other is the fact that in living systems you have problem solvers all the way down. In other words, there are systems all the way down that are not just mechanically doing whatever they do, but they actually do have a homeodynamic and other problem solving capacities. And that works. What that ends up doing is, first of all, one of the amazing things about the decompression side is that because you've lost information during the compression, it means that the right side of that bow tie has to be creative. I tend to think that the left side, the compression, can be algorithmic, but the right side has to be creative, because it's kind of like on the cognition end, it's the same thing. We don't have access to the past. What we have access to are the engrams that were made by previous versions of you in your brain and body. And then it's your job every, 300 milliseconds or whatever it is, to continuously interpret those things and build up an ongoing, a labile story of what you are, what you're doing, what's the outside world, what do your memories mean? And in doing that, it's basically a semiosis, because these are messages left for you from your past self. They're just like messages you get laterally from your, from things that are going on at the same time. And it's this process of building the best story that you can at any given moment, not necessarily the story you used to have. And that becomes really interesting in development, because if your allegiance is not to the fidelity of the interpretation, meaning I need to do it exactly the way my ancestors did, but instead what I think it's doing is I think it's a problem-solving agent that says, here are the prompts that I've been given. What is the best thing I can put together right now? And that results in some amazing plasticity in biology. I'll just give you a couple of my favorite examples. This Fankhauser work was incredible. So you got this newt, and you take a cross section through the kidney tubules, and you see there's like 8 to 10 cells that work together, and they give you this thing with a lumen in the middle, and it's this nice tubule. So you can make these polyploid newts. If you make polyploid newts, they have extra copies of the genetic material, and as a result, what they do is they make their cells bigger to accommodate the bigger nucleus. Okay, so that's cool, but the newt stays the same size. So you say, well, how did that happen? Then you take a cross section and you see that actually, well, then there are fewer of these larger cells to build the same thing. If you really go all the way up and you make, I think there were like 6 N newts or something like that, you have one gigantic cell that bends around itself and leaves a hole in the middle. Now that's a different mechanism. That's cytoskeletal bending instead of cell-to-cell communication. And just think about what it means if you're a newt coming into the world. What can you count on? I mean, you can't really know what your environment's going to be, but you can't even count on your own parts. You don't know how many copies of your genetic material you're going to have. You don't know how big your cells are going to be, how many cells, as Cook showed, you can, take away cells, add cells, it doesn't matter, it'll still rescale. You have to be able to do the best you can under whatever circumstances you find yourself. And in bioengineering, we see this all the time. Life is incredibly interoperable. You can do these crazy things, as you know, chimeras, and you can engineer all kinds of novel materials and scaffolds. This is one reason I love this. So we make xenobots, we make anthrobots, which are these human cell derived biobots. They have these incredible features because it will work like hell to get to the species specific target morphology that's the default. But if it can't do that, it will certainly put together something else that's coherent and that has, these anthrobots, no change. We don't touch the genome. So exactly normal wild type genetics, they have 9,000 differently expressed genes in their tissue of origin. They're in the same kind of milieu, they're in the same, but they have a different lifestyle. They're these little ciliated things that run around and do interesting things. Half their genome is now expressed differently. Xenobots express about 900 genes differently. They express a cluster for hearing, and so we've actually tested that. And it turns out that unlike frog embryos, these Xenobots can actually respond to sound, and so they have this new lifestyle. So I think this creative aspect, the constraint is it's kind of like a meta constraint. The constraint is you can't count on the past, and you can't take literally. You need to take the messages you get from the past, both your own memories and the genetic, experience of your ancestry. You take it seriously, but you don't take it literally. And as a result of that, because you can't count on the details of the past, you have to commit to a creative interpretation, which makes for incredible plasticity. It's why these tadpoles that Doug Blackiston in my lab made that have eyes on their tails, they can see out of the box. You don't need new. The eye doesn't connect to the brain; it connects to the optic nerve. The optic nerve connects to the spinal cord. No problem. No new rounds of selection and mutation. No, they can see out of the box. So all of this amazing plasticity is because they could never depend on doing it this. I mean, maybe C. elegans can. I don't know. Maybe nematodes could or something. But most things they're going to find themselves in all sorts of novelty, and they never assume it was going to be the same. They have to solve the problem each time. And that when we see this in the evolutionary simulations that we do. Because what happens is, as you start simulating this kind of unreliable and yet competent material, what happens is you have some mutations, but the material tends to fix it, and then selection can't see the genome as well because the phenotype doesn't show evidence. You fixed it, and so when selection can't tell whether your genome was great or actually your competence was good, all the work of that evolutionary cycle starts being put into making the algorithm better. And the more you do that, well, the harder it is to see the genome. And so you've got this feedback loop. It's this like positive feedback loop where over and over again, what you're getting really good at is doing something coherent, even if the starting material and the starting information is different, degraded, unreadable, whatever. And then that's how you end up with things like planaria that have a ridiculously noisy, a genome that's mixoploid, where every cell has a different number of chromosomes, and yet they're the ones that have this like incredible cancer-resistant, no aging, highly regenerative animal, even though the genome is basically junk. So yeah, I love the interpretation of that aspect of it. How to interpret that information.
Right. So I like to think about this also as looking at the hypothesis generation side of things. That in effect, that's what's going on, is that you use the same old information, but in a different context, you generate a hypothesis. The hypothesis already starts with bias. It's like this process we call Bayesian, but the hypotheses are actually quite sophisticated in biology, because you've already got a system that evolved to be confident in multiple ways, that evolved to be able to interpret under lots of diverse conditions. I used to tell my students, when we're talking about development of the nervous system, that nervous systems always, in a sense, adapt to the bodies they find themselves in during development. You add an extra eye, it adapts to it. You move the eyes, but in those of us who are mammals that have eyes, sometimes to the side of our head, sometimes towards the front of our head, it turns out that the way that system develops in the visual cortex, in effect, is an adaptive system. It's taking in information and saying, use that information to guide how it's wired up. Adapt to the fact that no two individuals will have eyes in the same relative position. And so the system couldn't work, it couldn't evolve if it had to be fine-tuned by virtue of a kind of deterministic wiring. It has to be adaptive in order to work for precisely these reasons. But that's in a sense, the decompression side of things. The decompression side of things has to itself be a kind of evolutionary process.
Yeah.
A hypothesis generating testing hypothesis.
Yeah, something that we recently did, and this preprint will go up, I think, this week. Is we made some Xenobots and we stuck some neural cells in it, such that what is the structure of a nervous system that doesn't have a selection, a history of selection for a specific purpose. So they find themselves in a completely new context. They're sitting next to cells, that shouldn't be there. What are the nervous systems going to look like? And these things are incredibly plastic. And because of this, because of the material has this ability to interpret and reinterpret information, you get these really interesting competitions between interpretations. So one of my favorite examples is in the early embryo, one of the things that tells the eyes where they should be, there's a particular bioelectrical pattern in the nascent face that has these spots and say this is where the eye goes. So if you inject one of a number of channels into some other location, you set up a similar bioelectrical pattern, those cells will get the message and they'll make an eye. But what's really interesting is that there's actually a competition, because if you section those eyes, what you find is that there's a few cells that we injected, and then they secondarily recruited a bunch of their neighbors to participate in making that eye. We didn't touch them. The cells we did inject broadcast there needs to be an eye here, and then so many cells need to sort of cooperate. But sometimes you do that, and sometimes you get no eye at all. Because what's happening is there's a competing interpretation which is a cancer suppression mechanism, where the surrounding cells are saying your voltage is wrong, and we're gonna equalize you out to us. They make gap junctions and they try to equal. So you get this battle. So these cells are saying no, we're gut, and these other cells are saying no, you should be eye. And it's the degree to which we are good or not good at providing convincing interpretations via these voltage gradients that we can specify a successful eye, or something that you can even see. Sometimes you can use animals that are transgenic for like early eye genes, like RIX1 or PAX6 or something. And so you inject your RNA and you see like there's eight ectopic spots. And you say, oh, we're going to have a tadpole with 8 eyes. Well, what you find out is that a bunch of them get winked out by their neighbors, and you don't get those. And then some, if you do it right, some of them will. And so that battle of interpretations, I think, is also really interesting, because that material, it's not passive matter. It can support things like this, like a debate between different anatomical worldviews about what are we building here,
Jerry, anything you'd like to add to that?
So much, but I'm not sure I want to keep going on this. And there's so many pieces. One of the things that when I think about cognition, there's a piece that I think that Michael was involved in terms of talking about getting the biology back into the notion of cognition. I have often thought that the way that we misunderstand brains is that we think of them in too much of a computational-like sense. When I think that brains are actually doing what embryos do, that brains are doing what evolution does, that we want to look at a thought process as, in a sense, what's sometimes described as microgenesis. That is, it's, in a sense, you're differentiating something as opposed to activating it or inactivating it. And the differentiation process, we experience it, as, new ideas come to fruition, or when we're sitting here talking and a new idea pops up, we differentiate it. It starts out pretty undifferentiated. And one of my approaches to language is to think about, in effect, what's a sentence before I've produced it? Well, it's undifferentiated. It's not that I go about and I say, okay, there's a bunch of rules and I want to pick out a bunch of, rules and positions and words. No, the words don't come first. The words are at the very end of the process. The sequence is at the very end of the process, and it's flexible. Thank goodness that I can move things around and it still works. In effect, we think about cognition too much like we think about reasoning, in a formal sense, when it's in fact, biology uses the same strategy wherever we look at it. So one of the ways I like to study different functional regions of the brain is to, first of all, look at how that region developed its circuitry. Now, a lot of it's Hebbian in the sense that there's a statistical logic to it. But actually, if you look at how different regions utilize the information that they're getting, the over connection that they received and so on, and the difference in connection from different areas, and how they settle it, neurons don't change their strategy when they become mature. They're doing the same thing at all levels. So they're, whether it's a nervous system just getting started and starting to wire itself up, as a result, the best way, from my perspective, to look at what a particular region of the brain is doing, what this class of neurons are doing, is to ask the question, how did they get that way in the first place? What developmental process was each neuron involved in? How did it amplify and eliminate certain connection patterns? How did it, adapt to the diversity of time frames that are coming in? All of those things in development determine how its circuit is developed. But I think that's what's going on, in you and I right now, that each of these is sort of you might say, a microembryology, a microevolution. Each thought that we produce. But then what we were talking about in terms of this sort of compression, decompression, creating memories is the compression process. We strengthen certain synapses and weaken certain others in order that when we throw information back into the system, we put metabolism back into the system, it will bias the dynamical pattern that's the result. But what's going on is the interpretation, or you might say the decompression, is going on dynamically. So I think of it very much like I think about the evolution of compressed signals in evolution, compressing it into DNA, creating what's sometimes been called the engram, is basically this highly compressed difference in synaptic strengths over a network. That's very, very different than how we do computing. And yet it's, of course, accomplishing something similar. But it's doing it, in a sense, it's biologizing cognition. And so I know Michael likes to talk about cognition all the way down. I like to talk about biologizing all the way up.
Mike, anything you want to say about that?
Yeah, I'm on board. It makes sense. These are kind of symmetrical views that we have here.
Terry, is there any part of Mike's work that when you first read, you found particularly different from your own, and that you questioned, and that you'd like to perhaps ask him about?
Oh, good. That's a good question, because some of the earlier work troubled me, and particularly with planaria. Planaria are a very unusual kind of animal because they're weird genetics, but also because of all of these things that we see in many plants, where a part of a plant can generate the whole plant, but we don't see that very often in animals. Mostly simple animals, Hydra and stuff like that, have these capacities. But as we get more complex animals, it becomes harder and harder, except for parts, parts of salamanders, parts of some frogs, and so on. They can do this. And of course, even some of our parts, but minimally, and they've become minimal. But I was worried that initially, particularly talking about the electrical and ionic systems, that it initially sounded a little bit like what I would call preformationism. That it initially sounded like something that a man who I never liked his work, a guy named Rupert Sheldrake, he had this idea that there was sort of, you had a field for a final map, that everything just sort of, if it disappeared, there was an electrical map of how things were supposed to go. At first, when I read this stuff, I was troubled by that. And I thought, no, this preformationism is not what we're talking about here. But in a sense, reading further and seeing how this stuff is developed in Michael's work, it's very clear that it came back towards this sort of decompression, reinterpretation logic, that it was not a freeform map, that there was not an electrical preform map here. I, of course, in working with the development of the nervous system initially, did not have any of the ionic information to deal with in studying it, because I was studying, the nervous systems of rats and mice and monkeys, and eventually people. And so there I was looking at sort of macroscopic features having to do with, how axons find their targets and things like that, mostly chemical and material processes. And from that, it was very clear that there was not, although things found their targets, went through and, interpreted signals and, in a sense, developed three-dimensional complicated structures, there was not an initial map. There was this compressed system of biases that set up another higher order physical process that was interpreting those biases and decompressing it. But in hindsight, when I look back at this early work with planaria, I realized that there wasn't quite this map. And I particularly like the later stuff you've done, Michael, where you've cut them in interesting ways and produced, sort of star-headed planaria and two-headed planaria, where you can see that in fact it's not a preformed map, but it's a complicated interpretive process that you can bias the interpretation in interesting ways by sort of critically doing it. So in this respect, I was wary of the early work and have recognized that it converges really well with how I like to think about these processes. But I'm particularly curious about the relationship of planaria and hydra to plants and how they generate from apart the whole.
Yeah, that's a very, very good point. So let me talk about this business a little bit, and I'll just preface it by saying that it is absolutely an interpretation and decoding process. I'm not saying that there is a preformed map that sits there as it is. However, if you zoom into the center of that bow tie, if you take a slice of it, there are time periods where the bioelectric pattern acts as if it was a map. And I'll just give you a couple of simple examples. And it's not just planaria. We've done this in frog, and we're now doing this in mice. This is I think a general thing. So let me just describe what we were looking for in what I was looking for is a physical encoding of a set point for this homeostatic process. So the thing about planaria and maybe sometimes I think that all development, like even mammals, half of us can regenerate an entire body from one cell, right? That's a kind of regenerative process. You're down to one cell and you can sort of reinflate back into the body, right? So, and we know that all of these systems will work like hell to get to where they're going. Even if you perturb them, you can cut them into pieces, you can move things around. We can make a Picasso frogs where everything's in the wrong place and they'll still kind of go back to where they need to go. What I was looking for is the encoding of the set point. Not magic, but nevertheless, any homeostatic system has to have somewhere. You have to have a memory of what it is that you're reducing error against, right? There's some sort of error minimization scheme. So I was looking for that. So it turns out that both in planaria head-tail decisions and in the frog face, if you look very early, before all the genes come on that are going to regionalize the face and all that, there's so let's just talk about the frog face for a second. There's a bioelectrical pattern which is readily decodable. And there are many other patterns that we are working hard to decode, and some are very hard. But the frog face one, it looks like a face. You can tell exactly here's where the eyes are going to go, here's where the mouth is going to go, here where the placodes out to the side are going to be. And sure enough, if you move any of those voltage states, not the cells, the voltage states, and we can do that with optogenetics and with other tools, if you move any of those things, the gene expression follows and the anatomy follows. And so you can, and in the planarian, it's the same thing. If you cut a fragment and you say, how does this thing know how many heads it's supposed to have? There's an actual voltage pattern that says one head and one tail. I mean, now we know that that's what it says. And if you artificially change it to say two heads, sure enough, you get a worm with two heads. And And parenthetically, if you then start chopping those worms, you forevermore will get two-headed worms, even though the genetics are still completely wild type, right? So for the duration of that homeostatic process that says, what am I supposed to be reducing error against? There is, in fact, a pattern that has a very important feature. The feature is, I don't have to worry about, like with any good homeostat, I don't have to worry about the hardware of how it does it. If I understand the encoding of the set point, I just change the set point and then I take my hands off the wheel, and the system autonomously builds to that set point. So for that period of time, I think you are looking at a map that isn't some eternal, static thing that sits there, but it is the set point against which that drives error minimization. Now that pattern has to emerge from prior states, and then eventually disappears. So it is not a permanent thing that just sits there, but it's sort of that center of the bow tie. It's the previous events have given rise to, at this moment, we're building the face. Later, we're going to build some other thing. It gives you the set point that guides the behavior, and that set point is really convenient. It's convenient because if you try to stick with, the standard story of development that we were all taught is that it's an open loop complexity emergence process. There's a bunch of local rules that everything goes according to the local rules, and eventually it's like voila, emergence, and you get this complicated thing. And yes, that can happen, and we know, like a cellular automata and other things, you can get complex things from simple rules. But that's not what this process is actually doing. If you start to interfere with it, you will see that it works actually really hard. It's not an open loop process. It tries really hard to get to a particular outcome. So that means, and the thing that's really hard about the standard story is that if you wanted to do, let's say, regenerative medicine, and you wanted to, this is the wrong outcome, I want a different outcome, or like what Terry was just saying about evolution, like going backwards, right, and figuring out how to encode it. It becomes really hard if all you have is this feed forward. That inverse problem is not solvable in general. But if you have a component where what I have is a self-organizing pattern, it's self-organizing because of some math and also because of the kinds of ion channels that evolution has provided us with. So it's an excitable medium, and then of course also Turing patterns of chemical signals and biomechanics and everything else. The math will give us a set point that for a period of time will allow us to get to a particular point, and serves as a really tractable control knob for when you want to make two-headed worms or fix birth defects. So that's one of the things we work out now is a very simple stimuli that repair very complex defect. For example, notch mutations, right? If you mutate notch in the brain is just completely wrecked. But the first thing that gets wrecked from that is the bioelectrical pattern that dictates the shape and size of the brain. And if you force the correct pattern, you can have perfectly normal tadpoles with behavior indistinguishable from control, like learning. They have learning indistinguishable from controls, even though that dominant notch, overactive notch mutant is still there, right? And so for a period of time, you get access to that set point that is very convenient. But of course, it's not just sitting there the whole time. It comes and it goes, and very much it's this inflation and deflation process. And what you're looking at is that bow tie node, that snapshot of that process.
Yeah, I like that because, and that's the differentiation logic. That is, you start from, and I'm thinking about the development of vertebrate embryos. You start with a head, tail, dorsal, ventral distinction. Then once that's set, now you can partition it into segments. Once those segments are set, you can now take segment A and recut it in a different way. Whether it's in gene expression in terms of polarities of things and so on and so forth. But it allows differentiation. Differentiation always involves this sort of sequence of steps, like you say, these sort of the center of the bow tie, where basically it's got a point where now this has been accomplished, I can move on to the next differentiation, shut down the old system, and now once it's set there, and I've got all of these divisions, dorsal, ventral, for example, division set, I can now do something else. But it's a differentiation process that begins undifferentiated and then becomes progressively, step by step, differentiated. And each of those is a decision like you're talking about, a sort of a node of decision. Now I can do this because that's been accomplished. But this is also the case that then if you mess up one of the early ones, the later ones have to do compensation to deal with what was modified in the first, but as a result, you can get, because the differentiation process, as you're describing it, sort of like a telescoping process that amplifies something. It can amplify something in a very different direction as well. And I think that's one of the other things that we're finding interesting about all of these processes, because this is that generative side. And this is why I think it's been so exciting in evolutionary theory to begin to focus on the generative side of things. It was originally described as evo-devo, but I think it's a much more subtle distinction than that. Evolution is taking advantage of both the compression and decompression side of things and how the differentiation can sort of be built upon prior differentiation. How you can start from like a single cell and develop a body. You can start with this is why I like to say that, in thinking, I have, what's a thought before I've differentiated it? It's a sort of, I sort of know where it's going. But it's not done yet. And like this sentence I'm producing right now, I sort of know where it's going to go. You sort of know where it's going to go. But it's undifferentiated. And the words just sort of pop in at the right time, thank goodness.
Mike, is there any part of Terry's work that you can articulate that you found particularly fascinating, but yet also disagreed with and think you could work on and build upon?
I didn't disagree with anything. The only thing, so I've been sitting here trying to remember what a good example of it. The only thing I guess we could talk about somewhere, and I don't remember where it was. You had talked about teleonomy as a kind of watered down version of teleology,
Yes.
and right. That's I'm remembering that correctly. So
Yes.
so the only thing I can say to that, and I think I take teleology very, very seriously. I think it's absolutely a real thing. I use teleonomy slightly differently. I don't use it as a way to sort of placate the folks who don't like actual teleology. Like I'm full on teleology. I think what's perhaps useful, and I don't know if this is one of these cases where I should use a different word or whether we should rehabilitate the existing word, but what I like about teleonomy is that it reminds us that what we observe is from the perspective of an observer. So when people say it's apparent teleology, I don't mean it's a lesser version or not real or some kind of like, not the real thing. What I mean is what I think it does for us, which I think is important, is to remind us of this, basically of this kind of intentional stance idea that it's not that it's not real, it's that you have to be an observer who's capable of seeing it. And finding teleology in things is an IQ test for all of us, because sometimes you don't see it, and it's not because it's not there. It's because we didn't know how to look or what space we were looking at or what the system was doing. And again, the system of a good agent will have its own perspective. It doesn't need our perspective, but nevertheless, we when all of these things, like I tend to think of a lot of these cognitive terms that people use as kind of as basically interaction protocols. What we're really saying is not that there's some objective, unique perspective, and we should fight about which one is right. But what we're saying is this is the formalism that I see, that I claim is going to be applicable to the system, and maybe it's some sort of high level planning, or maybe it's a simple, homeostasis or something in between. And then I'm going to interact with that system using this as a guide, and we'll all find out how well I did. And if you have a different estimate, we'll find out how well you did. So that's kind of the only thing that I found to even quibble about, is just that I think teleonomy has another use besides trying to water down teleology. It's just to remind us of this observer relativity of some of these things.
That's an interesting way of putting it, because my work has actually gone in a different direction in that respect. So thinking about the history of the term teleonomy, of course, it is an observer perspective. And the idea says that, they go back to Bigelow and Wiener and that group. And Pittendrich, when he comes up with the term, basically he said, we don't know and at the time it was, in a sense, even a stronger claim that there is no such thing as real teleology, that it's just sort of feedback and deviation minimization processes, and that rather than make a commitment to some metaphysical concept like teleology, the idea was that, come up with a description. Teleonomy is describing processes that are end-directed, accomplishing the same end from different origins, all the ways that people used it. Ernst Mayr did an interesting job of saying, but, there's different kinds of end-directedness in nature that maybe we should distinguish. So he came up with the idea of teleometry, teleometric processes. And he said the second law of thermodynamics is a teleometric process in a sense that it goes towards an end. It has end-directedness. It looks like it's end-directedness. It's not trying to get there. Things just tend to go in that direction. And he said that, but teleonomic processes, again, descriptive, and this is why your perspective way of thinking about it is appropriate. There's from the outside looking at something, it seems to be going in a direction, but it's a different kind of direction, he said. This is a direction that's error minimizing or deviation minimizing, and that we can see all that. And he said, but it may be, and he was not willing to make a commitment, that there are some things that actually are not just deviation minimizing. They use deviation minimizing, but they also have goals and ends. And the question is that when we design a system like a thermostat or a heat-seeking missile or something like that, the end is designed into the mechanism. But of course, with life, we're dealing with systems that create their own ends, design their own ends, design their own thermostats, sort of keeping my body warm. My body, the process is very homeostatic in the sense that it's constructed like a really complicated thermostat system. It's people could even describe it. It's not accurate, but roughly mechanistically in the trivial sense. But the fact that my body has evolved to have that mechanism, to set that value is like me building the thermostat to have a certain behavior, to have a certain end. The question is, where does that come from? And so this is one of the reasons why I think we are really forced to go back and talk about information about things. Because if teleology or teleonomy or teleometric, all of those refer to ends. But the question is, how do ends get established? How do they come into the world? That's been the problem I've been struggling with. And the answer is that you no longer can do this by the observer perspective. You want to ask the question, what kind of a system generates them? What's the architecture of a system that generates its own ends? And so that's been how I've approached it. And as a result, I've, in a sense, as I said early on, trying to do this thermodynamically, talking about how you move from systems that have the kind of end, the teleometric end that the second law of thermodynamics suggests, and then the kind of stable dynamical end that, say, Bénard convection or various self-organizing processes have. And then the kind of end in which the end is not just a pattern, but it's a representation, in which there can be a representation of that end, for example, in a compression, like DNA or whatever else. That's what evolution is about. Evolution is about generating that. But in that case, what I always found troubling about the teleonomic perspective is it always stayed at the observer perspective. I'm outside of the system, and I would describe its behavior, but not actually explaining how it generates its behavior. And so I basically wanted to approach this in the other way around. Ask, what kind of a system has these behaviors and generates something that I would feel comfortable of saying it's intrinsically about something, and that aboutness is something that it's not yet, that could happen, that might be the result of repairing damage and so on. So that's basically how I've approached it. And one of the reasons why I had a problem with the term teleonomy over the years.
No, I agree with all of that because the interesting systems have their own inner perspective on the goals that they have, which is not the same as what we see from third person observation. So a few of us, Josh Bongard and I, and Chris Fields and some other people have been working on very minimal systems to see what it takes to evolve exactly what you just said. So observers that where their various components are about something, that have that reference. And one of the things that we're finding is that, and this gets to the distinction that you just made around, there are goals that you give a system when you create the thermostat, and then there are sort of intrinsic goals that evolve and things like that. One of the things that we're finding, and there's not time to go into all the details, but I'll send you some of this stuff after. We're finding that there are very minimal systems, in fact, deterministic minimal systems, where there is no magic in the sense that there is no, in biology, there's always some mechanisms you just haven't found yet. Right? You can say, well, there must be something that you just haven't. So these things, we can simulate them. We on the computer, we know exactly what all the ingredients are. And what we see is that there are the things that it's doing because you made it do that, because the algorithm says to do that. But then there are also these weird side quests that the thing is doing that are nowhere in the algorithm. And these additional sort of intrinsic, these weird, some of them are what you would immediately recognize as competencies, things like goal, delayed gratification and things like that. But others are just new goals that the thing obviously pursues that are not specified in the algorithm at all. So I'm starting to think that ability to have its own goals that are not nailed down by the mechanisms or the algorithms or the materials or all the stuff that we're used to is really pretty baked into the world. And I mean, I think evolution does an amazing job at scaling that up to the point where we start, where it becomes obvious to us and that everybody can see that this is what this thing is doing, or even to the point where it can self-report like us. But at this point, my view is that that's actually, and in fact, Josh and I are writing a paper called "It Doesn't Take Much," which is basically all about these very basic, like very minimal systems that are already doing things that nobody told it to do. So, I think it permeates the world that we live in.
Well, Jed, it's we've got a hard cutoff soon, so
that's right.
Got 4 minutes left. I just want to say this has been amazing. It's been wonderful to be a fly on the wall watching the two of you. I'm surprised it's taken both of you so long to have a conversation like this. Any final words from the two of you? We've only got about 3 minutes left, so it's a quick one.
Please, Terry.
Wow. There's so much to deal with. And I would say that one of the challenges, and maybe it's a difference in the way you approach this, and partly it's maybe the result of Michael sort of coming from the computational world and me coming from this sort of Persian semiotic realm. But I'm really interested in this notion of self. And this notion that I've tried, what I call teleodynamic, a dynamic that actually has a represented end. By represented, I mean literally there's in a sense, a compression that will tell you whether you've received it or not, and that is not externally generated. But I think that requires self. And so I would say that the distinction between teleonomy and teleology in that respect is that teleology is semiotic. There's a notion of representation, and the problem for philosophy is that these are the most troubling concepts that we can think of. Purpose and directedness, self, sentience, benefit, cost. These are things that are just terrible for philosophical discussion. But in effect, we have now sort of crossed into domains where we're beginning to have tools to ask those questions. Something we haven't talked about. I think
we're close to the hard cutoff.
Okay, so the last thing I want to say is that we've never talked about it at all, but I think one of the things that's also bringing us to these questions is these changes in so-called artificial intelligence.
Yeah.
They're forcing us to re-ask these questions.
We'll touch on all of that. We'll try and have a round two soon. Mike, any final words from your side? Sorry, Ted.
Just to say thank you. To me, there's nothing more fascinating than this symmetry between the self-construction of minds and the self-construction of bodies. I think Turing already sort of saw that when he wrote that paper on on chemical organization, that this was two sides of the same coin. And I'm delighted that we're working on it from different angles. And happy to discuss again. Thanks so much.
And thanks.
Thanks, Michael. And thanks.
It was great. Yeah.
Really appreciate having you both on. But Michael, we know you have a lecture to give, so I think
I got to go teach. Yeah. Okay.
Teach. Cheers.
Thanks so much.
Soon.